In a previous lecture, we just discussed these two simplest models we can define for population dynamics, right? Namely, growth, when you don't have limitations, and death, right? Those models, if you remember, we use this simple diagram to in a way represent the pure growth process, right? This was associated with a simple differential equation which has a solution exponential growth, right? Remember, goes to infinity, but nevertheless it's useful when we consider the population where resources are not yet limited, right? The other process we considered was death, right? And for death, what we can use is a different diagram where essentially we say that this is a flow of the population to the empty set, in a way of losing population to zero, okay, over time, with a given rate. So what happened with other systems where both things are combined, which looks more like closer to the real world, okay? Well, one thing we can do is actually consider a class of processes that we're going to see again and again over different topics that combines two particular ways of increasing population and killing population, right? That's called linear growth models. The first one I'm going to talk about is a very simple model that involves two parts, right? One is the growth that we just, one is the death term we just described, and the other one is something that seems not very realistic, but you will see that it is realistic for some processes, which is the constant input process, okay? Constant input process means that we have a population X, okay, that receives at a constant rate individuals, right? Let's say at the rate mu, right? For immigration, for example, so that this process doesn't depend on anything, doesn't depend on how many individuals I have, it's just a constant rate of growth, okay? Very good. Just for the record, if you want to build a model of that, now that we have some idea how to do it, that's really the simplest differential equation you can define, because this is just the rate of change, so the speed at which individuals change is a constant. We just have an entrance of individuals, and this equation is very easy to solve, okay? If I had a given initial population, I just add more and more in a linear way, okay? Very good. You can actually check that by yourselves. It's a very easy exercise. What kind of models we want to address here? We want to address the following. Suppose I have a population X, right, that has this constant input, and you will see that there's a whole range of possible scenarios compatible with that, but individuals also die, okay? That would be my diagram for this. What kind of systems follow this, right? We'll see from the population dynamics to the molecular world, there are very good examples. Take, for example, this, right? You have trees in a forest, and they lose leaves over time at some given rate, right? I'm going to use mu as a constant rate of leaf loss. Of course, this is a constant relative leaf, or the species you consider, the time of the year, etc. But let's assume that, at least for a time scale, this is constant, right? So they dropped into the forest floor, and then the dead leaves decay, right? They decay over time at a given rate that I can write down as delta, okay? So I consider two different processes. Another example that is very interesting, and it's going to be relevant for a number of reasons, is something that you can do at home, right? Imagine you have some kind of vessel, okay? You have a constant entrance of water, which you can regulate, can be fast or slow, and you have here a sink. So the water that it's being stored here is also lost, let's say, at a given rate, okay, which is going to essentially be fixed by the amount of water you already have and the size of the hole that you have from which water escapes, okay? If you do that experiment, you will find out that no matter where you start, right, more or less filled, okay, given this rate, you eventually end up in a fixed level of water, right? The system gets attracted to that state. What other examples are there? I'll give you just two extra examples. One from physics, right? Imagine that you have a mass that falls, in free fall, okay? It falls following this force, right, which is constant, if you take into account that we are not very far from the Earth's surface, right? And imagine that you have moving in this direction, this will be the velocity, and you have air, the real thing, okay? Air imposes a friction that is proportional to the speed, okay? So, for example, someone is jumping in a parachute, it will not accelerate all the time until it smashes into the ground, but instead, because you have, on the one hand, a force that is constant and another force that is proportional to velocity, you eventually will also equilibrate. And an example that is especially relevant for us in terms of engineering biological systems. Imagine you have a gene, okay? This would be a very simplified picture of the promoter. This gene is transcribed into a RNA, okay? And this RNA degrades by some mechanism. Again, this mechanism could involve active processes, assume that it's just degradation, and assume that the gene is constitutive, meaning that it's constantly being expressed, right? That's again exactly the same kind of dynamical system. Why I emphasize these very different examples? Because it's very, very important when you do modeling to have in mind that once you reach a mathematical expression that is equivalent between very different systems, you can use anything you have derived from the mathematics from one system into the other. It doesn't matter if one came from pure physics and the other is from biology. Once the mathematics are the same, you reach the same mathematical conclusions. So, how can I do a model of this? Okay, that's a model that's easy to solve, right? We can just follow our intuition. If we want to express this, right, we could go into the discrete form. We just go directly into the continuous form. On the one hand, we have a contribution following this scheme, which is a constant, right? Very important to make a distinction. This doesn't depend on the population. It's just something that comes from the outside, okay? Minus, this is exponential decay, right? Proportional to the population. So my growth term here is constant, whereas my decay term, right, introducing death, is linear in x, okay? This equation can be solved, can be easily solved. Let's do it quickly. The following way. I can separate variables again, right? It's always nice when you can do that. The bad news is that most of the time these equations are hard to solve, right? But we will see how to actually not necessarily go into that. Okay, once I have x on one side, t on the other side, I can separate 0t x at time 0 for this particular value. And then what I'm looking for is for any arbitrary time, how many individuals do I have. I want the solution for this, all right? This is a simple integral. Well, the solution for this integral is a logarithmic function with the argument in the denominator between these two limits, x of 0, x of t. Right? For simplicity, let me take this one particular case. Let's assume that the original initial population was 0. I had nothing there, so I started to have incoming population, right? And this is just t. Well, the integral here is just t. I will move this minus delta to that side, okay, and use the two parts of this integration here for x of t, upper limit, for 0, lower limit, this is a 0, okay? And what I have is this, all right? Okay, from here I take exponentials from both sides. You know how it works, okay? I just give you the final solution. After some manipulations, you can show that the population of my RNA, of my amount of leaves in the forest floor, or the level of water that I have in my container, right, goes as mu over delta, right, for this particular initial condition, okay, 1 minus this, right? That's the equation. That's a solution for a system where constant incoming of individuals and constant death of the present individuals is at work. How does it look like? Well, if you do a graphical representation of that, okay? What we have is easy to show is we started from 0 for this particular case. It could be an arbitrary number, right? The solution would be slightly different. We grow and saturate. All right? That's the behavior of the system. Let's think about that a little bit. It could be 0, it could be 0, and this is when t goes to infinity, this term here, smaller and smaller and smaller, and essentially that's a nice part of this. When time goes, okay, this is going smaller and smaller, it goes to a constant value. All right? It saturates to a constant value. So it's mu over delta, right? Well, that's one first thing. Two processes of very different nature, one a constant, another depending on the population, leads into a saturation value. And an important part of this is that this saturation state is what we call an attractor of the system, meaning that what happened if I started in a different initial condition, not 0, but here, okay? I do exactly the same thing, and I saturate exactly in the same value. What happens if I started maybe closer to the saturation value? Well, there's less space to move here, but I also saturate into the same value. And what happened if, for example, I manipulated my system in such a way that I force the system to be in an initial condition higher than the saturation level, right? For example, I take this here, this is the saturation that they should achieve at equilibrium, I drop a lot of water there. No problem. The system just starts here and goes down, right? So that's what in nonlinear dynamics we call an attractor, right? Any initial condition, right, at least from a range of values, right, is attracted to the same value, okay? So that tells us something really interesting, which is that for a given values, for given rates, right, no matter what happens in the population over time, eventually I reach a level that is just depends on those rates, doesn't depend on the past story, right? Everything is attracted, and in a way, the memory of the past is erased, right? You go into the same exact value, all right? So, what kind of applications can we make for this kind of system for bioengineering? I just wanted to mention one thing, right? We want to, in a way, define the principles of biological design, right? And one interesting thing that we can ask ourselves, for example, is how do tissues regulate their size, right? How the tissues and organs are capable of actually knowing that they have the right size and do not keep growing, okay? The simplest model you can formulate, I'm not saying that it's right, the simplest model you can formulate is based on the following idea. And just formulate the idea because the rest, the mathematics is exactly the same. Imagine I have a tissue, right, that involves a number of stem cells, okay? I have a stem cell compartment, right? In our first approximation, we think stem cells in a population of cells that essentially is immortal, right? And essentially is more or less constant, right? There are several things that we should actually highlight here, but the first approximation, that's fine. And this compartment is responsible of creating the whole progeny of cells that eventually build tissues, right? In cell types for different tissues. So imagine a given type of stem cell, that's a constant compartment, and these stem cells, right, for example, a given stem cell, well, I'm using SC for population and for labeling stem cells. It's an abuse of language, but that's fine for me. Imagine that it replicates, and when it replicates, it gives another stem cell and some kind of cell type, right, which is differentiated, right? That happens again, right? And this cell population can replicate or not, it depends, right? But in any case, you see that the number of stem cells will remain the same, right? Now these cells here can replicate, maybe can differentiate into something different, etc. But let's make the simplest assumption, right? That these stem cells create some type of differentiated cell, right? These cells perform some functionality and then eventually die, right? This, for example, happens at some rate, eta, right? Differentiation into different. They decay, okay? And they might or might not self-replicate. We will go to that when we consider tissue architecture, right? And how to make models of that and why this has consequences for cancer. You can solve this model, right? That's an exercise for you, and show, and that's not a surprise now, right? That because it follows this kind of basic scheme and it has well-defined attractors, right? You will see that depending on R and delta, you will have an attractor or not like this, right? But it's part of the exercise. But you can actually say that you can have a design principle for a very simple kind of tissue architecture where essentially what I'm saying is what controls tissue size, how many cells of this tissue you have, parameters, just two basic quantities, right? So mathematically we can say that the final architecture in terms of the size of the tissue is an attractor of the nonlinear dynamics that, well, the linear dynamics that we have just formulated. Now the question is, what else can we do with that if we want to go a little beyond this kind of scheme where I have one compartment, single variable, right? And keep using these models into a context where I can consider two populations. Okay, so far we have seen models, linear models, right, where we have growth terms, death terms, immigration terms. If you want, in a very general context, we can think that the models we have been defining so far could be written in terms of a function that has this form in general, right? Could have linear terms, positive or negative, constant terms, right? There's an extension of these models that is especially important if you want to think in terms of biology, reversibility, and a very important process that is strongly dominating biology in many ways, which is diffusion, right? Diffusion happens at multiple scales also, from the diffusion processes that exchange molecules between cells, between the environment and within cells, crossing membranes. And in general, it's a problem that has an enormous relevance in both physics and biology. Let me give you an example of what can be said in terms of mathematical terms of the simplest diffusion process you can actually define. Okay? This process will involve two compartments, okay, 1 and 2, completely closed, nobody can escape from there, right? And for a change, let's assume that there's no biology here, just molecules moving in this part, okay? And at time 0, suppose that x of t labels the number of molecules in this part, I of t the number of molecules of the same gas or whatever it is in this part. At time 0, for simplicity, let's assume that x of t at 0 was c, a constant, the total number of molecules, right? Whereas I at time 0 was 0. No one was there, okay? We all know what's going to happen. As soon as I let this open, right? This is a connection that can be large, or with a large diameter, a small diameter, so making easier or more difficult to go from one place to the other, assuming that molecules here are moving at random, okay? But we all know what's going to happen, right? This is the arrow of time. If you wait, right, the molecules are going to get distributed more or less the same amounts in each compartment, okay? And that has a lot of implications, because in the real world it means that if you wait enough time for a process like this, with no other mechanism that counterbalance this, you end up in a state of maximum homogeneity, right? You lose information, you lose structure, right? Biology gets energy from the environment, right, to oppose this mechanism, right? Cells are not like their environments. They are distinct. And they are different because they actually make a use of energy of ATP and other resources to actually build a different environment within the cell, right? It makes a strong effort. And death at the cell level, in a number of ways, it means that those mechanisms that maintain this difference, right, get lost. I mean, they disappear. You lose the capacity for making a distinction between inside and outside, right? That's kind of the basic abstract scheme following Mary Shelley's idea, no? What can we understand about death? Well, that's one first step of it. So if you wait enough, what you see is that there's a homogenization. You don't observe the contrary, right? You don't see that, right? That's why when we see a movie of someone jumping into a pool, there's no surprise at all. We see a movie reversed and somebody going out from the pool, it sounds kind of strange. It's not normal, right? That doesn't happen, okay? There's a lot of physics here to discuss, but it's not the topic for this lecture. So how can we make a model of this, right? The process of homogenization, right, that we know that ends up in half molecules here, half molecules here. I don't have to solve the equations. You can tell me that that's the final state that I'm going to see. Half here, half here. So how can I show them mathematically and how the system approaches this state? Okay. All right. Again, it's not difficult to build a system of equations, but it's a system. Right? It's not a single equation. So it seems that I'm going to abandon the formalism that I used, but that's not going to be the case, right? The important thing here is, apparently, I don't have one equation, I have two. One for x, one for y, right? So how can I deal with that? Let's go into the intuition. There's a whole formalism to build what's called the diffusion equations, but stay with me with that, right? Imagine that somehow how likely is the process for happening, how easy is to go from one compartment to the other is a rate that I can capture with a single number, right? This, of course, includes more things than just a simple number. We don't go into the microscopic description, we just have this coarse-grained view. What I'm saying is, this parameter is telling me how easy it is to change, okay? Now the thing is, here, I need to put something, right, that has to depend on x and y. Somehow, I don't know how, right? So in principle, that would be a function here and another function here, assuming something very general. Well, but this has to take into account two things. One is that there's a flow between the two compartments, okay? So it depends on the amounts of molecules, at least in each compartment. The other, much more important, right, is I also know, that's a clear intuition, that when I'm here, right, the difference between this compartment and this compartment is huge. It's as large as possible, okay? The flow is rapid. Lots of molecules are going to move into one compartment to the other. Whereas here, or when I'm approaching this state, the flow in both directions keeps going, okay? And it's very likely that I'm not going to see too many differences, right? If this is slightly larger than this, it's going to be a flow, but the flow is going to be slow, right? Since this measures the speed of change, okay, that's my conjecture, right? This has to be something that depends on the difference of concentrations, okay? Why? Well, if there's no difference, because I'm already in this state, it's not going to be flow at all, right? If this is C halves, is C halves, nothing happens, right? Okay? So this should be 0, no change. On the other hand, if this is large and this is small, maybe I have some molecule, okay? The flow in this direction is going to be strong and negative for me, okay? The balance for me, if I'm in that scenario, is that I'm going to lose matter, whereas this one will gain matter, right? Here is my proposal. This has to depend on concentrations in this way, okay? See how clear is this. Imagine that, forget about this for a moment, imagine that y was 0, right? It's the biggest difference, right? No one here, everybody there, okay? So this will be negative, okay? And correspond to exponential decay, essentially. Everybody moves into the y compartment, the second compartment, okay? Whereas if y and x are similar or equal, right? This term is going smaller and smaller, or 0, it cancels. And the same happens here, except that exactly the opposite way. Okay? That would be the simplest way of formulating what is called the diffusion equations, diffusion model. All right? Okay, now you could say, all right, but we have to solve this system of equations, right? We just came from models with a single variable, unidimensional models. But in fact, it's not true. The reason is this is closed, completely closed compartments, so nobody can escape from here. So I know that if the concentration, the initial was C, everybody was there, this everybody is going to distribute between both parts in time. So at any given time, the total amount of molecules is going to be C, right? So in reality, X plus Y is something that tells me that there's a strong constraint, that if I know one of the variables, I know the other. It's not really a two-dimensional system. Once I know the state of one compartment, I know the other. So it's one-dimensional system. And if I use that, see how simple it gets. Let's take this equation here, right? All right? For the first compartment, I had this equation. But now, in reality, this is just this. So I end up with an equation that is already familiar to us. The equation of diffusion, one equation, for my system is just this. Okay? That's the simplest equation for diffusion that can be formulated mathematically, right? And I'm saying that you already know about this equation because if you look closely, this is a constant, right? Minus another constant by x, right? So it's not different from the equations that we just saw before of this kind, okay? And remember what you just said. Those equations refer to populations of individuals that have immigration, etc., etc. It's a different problem, but mathematically it's the same, right? We don't have to rebuild all the calculations, right? We can actually solve the differential equation, no problem at all. And if you do it, it's possible to show, the mathematics are exactly the same, that the system, for this initial condition where everything is in x, goes like this, okay? If you calculate the same for the other variable, for y, it's almost the same thing except for a sign, right? These are the solution. This is the solution for this diffusion process. If you want, can write down this in these two forms, right? How does it look like graphically? Well, easy. This is time. This is, let me put x this way. I have a limit here that I cannot overcome in any way, right? It's different from the population models we discussed before. I can force the system to have a larger value. Here is limited to a constant value. And I also just will see, as we knew, the intuition we had, right? That in the end, the system gets attracted into the half and half state, okay? For x, the equation goes like this, starts from C and goes with this exponential term to C halves, all right? My recommendation always when you solve the equations is to make a little test. For example, this is the solution when t is 0, right? This is 1, 1 plus 1 with this 2 is C, okay? That's fine. This goes here, and if you do the same for y, right? Y, when t is 0, this is 1 minus 1, 0, starts at 0, okay, and goes up to this value, right? Again, it doesn't matter where you start in terms of the balance between the two parts, provided that you always follow this, okay? This is an attractor of the system, right? The homogeneous state is an attractor of the system, okay? Well, as I was saying, the diffusion process is important for many reasons, right? One is that it's something that happens all the time across biological membranes, right? Yes, usually these lipid bilayers, right? And molecules, usually small molecules of different kinds, right, can cross from the outside of the cell to the inside of the cell, right? However, as I was saying, if that was all, there would be no biology. Right? No biology can be built unless you have compartments that properly separate the inside from the outside and make it different, right? And that is achieved because there's something else, of course, that in the middle of these membranes, okay, you have molecules, proteins in particular, right, that play a very different role, the role of allowing things to get inside differentially. You use energy to pump in and pump out things in such a way that when you look at the inside and you look at the outside, they are different things, right? Here you maintain metabolism, you maintain information, and you process everything. Here is the environment that you have to react to, okay? All right, how general is this, and what other things can you do with that? For example, there are many problems you can formulate in this way, and experiments that can be done easily. For example, consider you have a table. You have a table with coins. Right? Imagine that I put all the coins face up. Right? Right? If you want this, all heads. All heads. I have all heads. Okay, this is a very peculiar state. Imagine that now I make the table vibrate, okay, by just pushing it. Okay, the coins start to jump, right? And what is the final state of this process? The final state is that on average, right, half coins will be up, half coins will be down, right? Think about how you can formulate that kind of model. You will see that despite the apparent differences, there's no two compartments, there's no something like a diffusion value, etc. It's mathematically essentially the same process, right? So that's for the linear models. And now the next step that we need to face, and that's what makes biology and in general complex systems interesting, is to see how nonlinear phenomena are going to change everything that we just showed here, right? When I'm here, right, the process of diffusion is a linear process, essentially, okay? The process that are going to make a difference by pumping things in and out is nonlinear. And the requirement of nonlinearity makes some of the mathematics more difficult, but surely much more interesting than what we have been seeing so far.